All right, this is going to be part two. We're going to talk about antimicrobial action. And I have created this table for you. It really simplifies. There's a lot of information in the textbook. So this is basically what you want to be able to remember or to know about for the exam. It's still kind of a lot, but we want to know the target of the bacteria. So what is it that we're targeting of the bacteria for these antimicrobial drugs? What is going on when we target the cell wall or the nucleic acids? An example of those drugs. So we have penicillin, we have fluoroquinolones, rifampin, polymyxin B, tetracycline, sulfonamides. These are, hopefully you've heard of some of these before. I know penicillin, maybe we talked about tetracycline at the very beginning. Sulfonamides are also known as sulfa drugs. And then we have a here where when microbes are resistant to these drugs, right, what do they do? So we're going to move through these slide by slide, starting with an image of the targets again. And we have the cell wall, right? So again, this is a selectively toxic target because we don't have cell walls, and the molecule of peptidoglycan is unique to bacteria. There are lots of drugs that attack the cell wall, right? So we're only going to talk about we're only going to talk about penicillin, which happens to be in the category of beta-lactam. We'll look at that, how it works. We can target the plasma membrane. There are some molecules within the plasma membrane that we can attack. Polymyxin B is what we're talking about there. Let me just make a pen, right? So we have beta-lactams and penicillin. We're going to talk about polymyxin B. We can also attack the translation process. So we talked about how the ribosomes are they're structurally different. And I chose tetracyclines as an example because this is something that hopefully many of you have heard of. Moving on down to metabolic pathways. These are the enzymes. These are unique metabolic pathways that bacteria have that we do not. So we want to target that as well. So we're going to talk about folic acid synthesis, and this is the sulfonamide. And then we have DNA synthesis and RNA synthesis. So I know that we also have DNA and RNA, and our DNA and RNA is the same. However, the enzymes that copy DNA and the enzymes that make RNA are slightly different than ours. And so we can target those as well. So for DNA, we have the fluoroquinolones, and for RNA is rifampin. So we'll take a look at that. We're going to start off with, which I didn't think I was starting off with. Okay, let's start off with DNA and RNA synthesis. So here is, if we remember back to our chapter on the genetics chapter, we have DNA replication, where DNA is copied. And then we also have transcription and translation where DNA is expressed. So this is talking about both of those. So if we want to target DNA replication, we're going to target an enzyme called DNA gyrase. DNA gyrase is actually unique to bacterial DNA replication. Humans, eukaryotes don't have gyrase. And the reason is because bacterial genomes, as you remember, are a circle, and eukaryotic genomes are linear. So I'm not going to get into too much of the details, but this really helps with replication when you have a circular piece of DNA. And it just targets that. So if you create a mutation, or so you stop this DNA gyrase from working, you're going to stop the DNA from copying itself, and so therefore the bacterium cannot divide. And then we have our RNA polymerase. So this guy here, this big gray molecule, is going to be the one that's going to open up the double strand of DNA. It's going to read one strand, right? It's going to create a molecule of RNA that's a complement of our strand. RNA polymerase in bacteria are slightly different. They're slightly structurally different than eukaryotic RNA polymerase. So there's certain target sites on this enzyme that can block transcription, right? So transcription, remember, is the process that creates the messenger RNA. And then this is our drug here, and these are our drugs here. Okay, so just real quick, we're stopping gyrase, and we're stopping polymerase from working. The next up here is our plasma membrane, our polymyxin B. So the plasma membrane, as you remember, the function of it is basically a gatekeeper for all molecules, right? It's a selectively permeable barrier And when it's disrupted, you can cause leakage of all kinds of nutrients. The cell will, lose water. All different kinds of mayhem will go on. So the membrane is disrupted with polymyxin B. So I want to just show you, this is not something I'm not going to require you to memorize this. However, what polymyxin B does is it inserts itself into the phospholipid bilayer. And so there's two examples here, right? This is going to be our example of a bacterium that is going to be resistant to polymyxin B. So you can see that there's actually two lipid A's. You guys remember lipid A? Lipid A was a part of the lipopolysaccharide, the LPS molecule. So for some review, LPS molecules are only found on gram-negative bacteria, right? Because it's part of the outer membrane. And E. coli happens to be a gram-negative bacteria in this example. And so if we look at the two scenarios, right? So this membrane here, if we look at the two lipid A's, one has a red dot because it's a modified version of lipid A. Through mutation, some bacteria have this modified version of lipid A. If you have the modified version of lipid A, look what happens. The polymyxin B can't insert itself into the phospholipid bilayer. If you have the normal lipid A, polymyxin B drives itself and disrupts the membrane structure so that if you're resistant, nothing happens to you. However, if you are susceptible, you have the normal lipid A, you're going to leak all of your nutrients, all the water out, and you're going to have cell death. So, a snapshot of how the form of the target, like the form of the membrane, those small variations in the membrane can cause an antibiotic to be useless or, kill the bacterial cell. Now we get to the beta-lactam rings. So this is going to be your penicillin. These, if we look at the class of beta-lactams, penicillin, cephalosporin, this guy, they all have the same sort of central ring here that is going to be the same. And then the other parts, which we call the R groups, are going to be different. What that beta-lactam ring happens to do is that it can, I thought I had a picture, never mind. All right, what it does is it can disrupt the enzymes that make peptidoglycan. Really simple. It prevents the cross-linking from happening. If you remember when we learned about the peptidoglycan cell wall, there's NAG-NAM sugars, and then there's a protein cross-link that holds those sugars together. And this inactivates the enzyme that makes that cross-linking happen. So the downside of these beta-lactam rings is that bacteria have evolved an enzyme called beta-lactamase. Okay, this enzyme basically comes here and destroys our antibiotic. So it can recognize that beta-lactam ring and it can destroy it. So these guys all share the same nucleus of the beta-lactam ring and different R groups. So depending on the enzyme, depending on the beta-lactamase enzyme that the bacteria have, it might be able to destroy penicillin, but it might be susceptible to carbapenem. I don't know how to say these words. I haven't said them out loud now that I'm noticing. But anyways, so sometimes you can try that. Like you can just try a different, it's like the same target, but it's slightly different in its nature. So it might be able to work on the enzyme that makes the cell wall, and beta-lactamase would not destroy it. Okay, so again, the bacteria make beta-lactamase that destroys the beta-lactam ring. Beta-lactamase is an enzyme, so it's, the active site's pretty specific. The reason why we have different forms of this is because those R groups can sometimes not make it so it won't fit into the beta-lactamase enzyme and it won't get destroyed. All right. Our next one is going to be protein synthesis, and this is going to be our tetracycline. an image just to have you see that the ribosome, we have the 50S large subunit, the 30S small subunit. I know it does not add up to 70. It's based on a different sort of, I don't know what to say, experiment or a different property than weight. And so basically we're just stopping translation. So remember, the process of translation is to read the messenger RNA, right? So this is our RNA. We're adding the amino acids within this unit with the ribosome. And the different drugs here just stop that process. So we don't need to memorize the details here, just knowing that it can bind to either subunit and it disrupts the addition of amino acids, stopping protein synthesis. Our next topic is going to be our metabolism. So we're going to look at the process of making folic acid. The drug is going to be our sulfonamide here. So turns out folic acid, which is a B vitamin, is something that bacteria can make. Okay, they can synthesize folic acid for themselves. It's necessary for survival. We do not make folic acid. Folic acid is something that we need to get from our food in our diet. So we don't have this pathway. We don't have this folic acid creating pathway. What the sulfa drug is called an anti-metabolite. Okay, so you see that word bolded here. And it stops the process of making folic acid, which stops bacteria from growing. So let me just explain what's happening here. So we have this PABA molecule, which is natural in the bacteria. We don't want to worry about everything here, but PABA is the substrate. This is the enzyme. And when the substrate and enzyme come together with glutamic acid, they make folic acid, right? So this is the chemical equation. The enzyme is this guy. If we have taken a sulfa drug, the sulfa drug looks just like PABA. It looks just like the substrate. So it's going to attach to the enzyme's active site. The enzyme is fooled. It cannot make folic acid. Okay, so we're basically disrupting the process of folic acid. Think of this as a competitive inhibitor, right? It's going to compete with PABA for the active site of the enzyme. And therefore, if we block the synthesis of folic acid in bacteria, they're going to die. Or they inhibit growth, they inhibit growth. All right, so that was antibiotics, right? So against bacteria. What other antimicrobials are out there? We have anti-fungi, protozoan, and worms are helminthic targets. We're going to go through each one of these. This is another chart for you guys it's a shortened, abbreviated table. And then viruses are also, we have to talk about antiviral drugs. So the first thing we're talking about is the antifungal. We've looked at this word before, ergosterol. Ergosterol is only found in fungi, and it is very similar. It is a cholesterol, okay, but it's a different kind of cholesterol. So we can see cholesterol has this 4, 1, 2, 3, 4 fused ring structure. It has these functional groups. We talked about these R groups attached. And if we look at ergosterol, very similar four ring structure, but completely different R groups, right? So we can create a chemotherapy that attacks just the ergosterol and leaves the cholesterol alone. And this is from your textbook. There's actually many targets of fungi that we can use and exploit. So I'm just going to go through them, right? We can disrupt microtubule function. We can stop RNA and DNA synthesis. We can inhibit another mitochondria from producing ATP. We can also inhibit the production of these sugar groups called glucans in the cell wall, inhibit other structures within the cell wall. So chitin, remember that word chitin? That was the material that the fungal cell walls are made of. That's a nice target for them. But I'm talking about here the ergosterol. So we can see how ergosterol is really part of the membrane. It's an integrated cholesterol molecule. And what our antifungals do is they just disrupt the production of ergosterol. And then if ergosterol isn't there, the membrane is going to be disrupted and not be good for the fungus. It's going to kill the fungus. Okay. We don't have images of, sorry, I keep flipping, but we don't have images of antihelminthics or antiprotozoans. But if you look at the mode of action, we're going to, you guys are familiar with this now. An antimetabolite is going to be something that disrupts an enzyme of a metabolic pathway. We can stop DNA synthesis or inhibit the electron transport chain. It can bind, so antihelminthic for worms, we can bind to ion channels. And for worms, now we're talking about a big animal, whereas fungi or protozoans are either unicellular or, very small. But we're talking about a worm here that can be huge. So it's going to be a little bit different attacking them. So we're going to block some of their ion channels that block the nervous system from working. And it causes the worms to either starve, or they're paralyzed. And so what that does is it basically unlocks their jaw, like their scolex, for example, on tapeworms. It'll release from the wall of your large intestines, and then you can just sort of eliminate it. And sometimes you can cause death of the worm. I wanted to put the example ivermectin here because it gained so much notoriety during COVID. Remember, people were taking ivermectin to get rid of a virus, COVID, which has, if you look at the action of ivermectin, what here is similar to a virus? Nothing. Okay. So if you look at the resistance, so the nice thing about ergosterol as a target, an example is miconazole. So this I think is also kind of common. If you get any antifungal over the counter, it's going to have one of the azoles. So azole is something typical you'll see for an antifungal, and miconazole is very, very common. There's not a whole lot of resistance to miconazole, so that's why it's always in antifungals over the counter. Fungi can mutate around it or pump it out. We'll look at those efflux pumps in the second part, or third part. There's the mutation of the target, also another pump. And then for worms, sometimes they just mutate around the drug. So the cells are not susceptible to those drugs. Okay, so we're going to move on to antivirals. Okay, so the first antiviral here is going to be called a nucleoside analog. Okay, so we know about nucleotides. Nucleotides, A's, T's, C's, and G's, are incorporated into DNA when you're building DNA. And acyclovir, this drug, is going to be a mimic of our nucleotide guanine. So it does say guanosine. Before it becomes a nucleotide is a nucleoside. So just a small little difference. Don't worry about it, but you just want to know that acyclovir is going to mimic the G, right, guanine. And if you took it, right, so let's review viral pathology. The virus enters into the cell. It will uncoat, right? And then what the virus is going to do is going to want to build its DNA, right? Viral enzymes add a phosphate group to acyclovir, and then human enzymes add 2 or more phosphate groups, introducing acyclovir triphosphate. So basically, when the virus gets in, the virus does some work. Again, not important, but we're going to create basically a really nice fake version of the letter G. And then when the virus wants to reproduce its genome, it, will take the drug instead of an actual guanine. So this is the drug acyclovir. This is the actual guanine. So it'll take this and incorporate it into the growing DNA strand. But this doesn't have an attachment site for the next nucleotide. So wherever the G gets put in, like it's going to get put in right here, right? Because there's a C across the double strand. That's going to stop. So we cannot add another nucleotide to the acyclovir because it doesn't have the groups to attach to. So it doesn't have the necessary chemical grouping to continue copying DNA. So it's basically going to stop viral DNA from replicating, right? So it's a structural analog to guanosine, and it's going to, once it's incorporated into the growing DNA chain, it'll stop replication. All right, there's a special therapy for retroviruses. So retroviruses An example is HIV. Okay. Retroviruses are viruses that have a negative strand RNA in their genome, which means that once the RNA enters into the cell, it cannot be used as messenger RNA. It actually has to be reverse transcribed back to DNA. And then the host machinery will then make the appropriate messenger RNA, and then that will go on and make protein, right? So the virus requires that the negative strand become DNA, and that's not a normal process that we have in the cell, right? We always go from DNA to RNA to protein. So this reverse process is unique, and it takes a special enzyme called reverse transcriptase, which is right here, reverse transcriptase. And it's so important to the virus's life cycle that the virus actually carries reverse transcriptase inside of its capsid. So one drug called AZT is a reverse transcriptase inhibitor, right? So it basically stops that enzyme from the RNA to DNA. We want to stop that process. So that's one target of antiretroviral therapy. If you do have HIV, then they usually give you a cocktail of different drugs, which is called antiretroviral therapy, or ART. And the targets in that antiretroviral therapy are all of these guys. So we're going to have an AZT within this therapy that's going to inhibit reverse transcriptase. The virus, the HIV virus, the retrovirus, also needs to integrate itself into the DNA of the host, and that's done by an enzyme called integrase. And then so we can have a drug that basically blocks integrase. So it's an integrase inhibitor. Then we have, once the virus gets made, it gets made in these large chunks, and then those pieces of protein need to get cut down by an enzyme called protease, and we can also inhibit that one. So we can stop the virus from really forming its subunits that it needs to build itself. So that's protease inhibitor. And then lastly, the last one here is going to be a fusion inhibitor. So there's going to be a mechanism that blocks the addition. So remember we talked about spikes on the surface of a virus. This is an enveloped virus. The spikes are going to attach to a receptor on the membrane, and this interaction can be blocked, and then they're called fusion inhibitors, okay? So understand the concept, right, of what an ART therapy is about. It's about blocking reverse transcriptase, blocking integrase, blocking protease, and blocking fusion, all to sort of halt the HIV from replicating. In just one word, the reason why we have so many targets at once in this antiretroviral therapy is because these RNA viruses mutate very quickly, and it is quite possible that the reverse transcriptase, the AZT, that the enzyme can mutate around it. So if this doesn't work anymore, then we still have several targets still in that therapy. All of these antimicrobials can have side effects on the host, on the person, right? So it's anything you take into your body, whether it's a supplement, whether it's an actual, manufactured antibiotic, it doesn't matter what it is, your body is going to have to clear it. And it can have unintended effects on the body. So even though you want drugs that are selectively target, chemotherapy, by its nature, just by taking something that's foreign into your body, can become toxic to these. It's toxic in your body in some way. 5% of all people have some serious side effects, and then the side effects tend to come into three categories. So there's some kind of toxicity to organs. The liver is a big one here because the liver does detoxify substances from your food, right? So you're eating some vegetables, they might have some pesticide on it, right? Your liver's responsible for detoxifying that pesticide. And then your kidneys are also a big one. Your kidneys will have to clear that, right, and put into the urine to get rid of. So those are really big, two big organs that are affected a lot by chemotherapy. Your nervous system can too. Some examples here that sulfa drugs can crystallize in your kidneys and form stones. And then chloramphenicol can cause severe or irreversible effects. Allergic reactions are also really common. I'm allergic to sulfa drugs. When you have allergy, okay, so the difference between toxicity and allergy is allergy is mediated by your immune system. Your immune system is looking at that antibiotic as a foreign allergen, right? And will cause this reaction to it. So it's basically an allergic reaction, rashes, itching, hives. And that can be also very severe, or it can be mild. But toxicity is the damage to the cells of the organ. Liver cells are being damaged, kidney cells are being damaged. Allergy is the immune system's reacting and causing these allergic reaction effects. The third thing here is disruption of normal flora, which is what we talked about in superinfections. It's the most common because when you do take an antibiotic, it will kill off, right, the intended pathogen that's making you sick, but it does also kill off some of your microbiome. It just does. And so when you upset the balance of the microbiome, diarrhea is a common symptom of that, right? And then this is an example of a weird side effect where when you take tetracycline and when kids take tetracycline and their teeth are developing still, they can actually cause this yellow pigment to be deposited, and it's permanent. You can't get rid of it. You can't like crest white strip your way out of that. It's going to be there for the rest of your life. All right, and that's part 2.